Hello, my name is Megan Lott, and I'm a third-year PhD student in the Department of Environmental Health Sciences at the University of Georgia. I work with Dr. Lipp in the Environmental Health Microbiology Lab at the College of Public Health at the University of Georgia. Recently, our lab has focused its efforts on monitoring the progression of the COVID-19 pandemic in our local area using a novel approach to disease surveillance, sewage monitoring. Here, we present to you our investigation of wastewater-based epidemiology in our college town. Wastewater surveillance is an emerging tool for monitoring public health. Wastewater samples are viewed as a composite sample that captures the whole of the community. Wastewater collected across neighborhoods, towns, and whole cities typically flow into a receiving wastewater reclamation facility. By collecting influent wastewater at the water reclamation facility, we can capture a snapshot into the health of the entire community. In this way, our largely unseen or forgotten wastewater infrastructure can emerge as a brilliant source of information. Wastewater-based epidemiology has been used for a wide range of applications. Previously, researchers have examined wastewater to monitor the use of prescription or illicit drugs at the community level. Wastewater has been mined for biomarkers related to other health behaviors, such as tobacco and alcohol consumption. The microbial and molecular characterization of wastewater has demonstrated that wastewater reflects population-level traits of humans, such as those indicators associated with obesity and other chronic conditions. Notably, wastewater-based epidemiology was used to monitor the progression and efficacy of the global polio vaccination program of the early 2000s. Targeted wastewater monitoring was used to detect circulating poliovirus before, during, and after vaccination programs within neighborhoods and across entire communities. This wastewater-based approach provides a new approach to infectious disease epidemiology. Sewage surveillance may be used to examine and predict the total burden of disease at a community scale without the need for extensive clinical sampling. Wastewater-based epidemiology, or WBE, has emerged as an effective way to monitor the progression of the COVID-19 pandemic by examining total viral levels of SARS-CoV-2 in wastewater. Since the emergence of the SARS-CoV-2 novel coronavirus, several key findings have underscored the applicability of wastewater-based epidemiology for monitoring community-level viral transmission. First and foremost, viral RNA of SARS-CoV-2 is shed through and detected in stools. There is little evidence that infectious virus is shed through stool. However, researchers have demonstrated that individuals with COVID-19 infections shed viruses in their stool for prolonged periods of time, sometimes even after the virus is no longer detectable through a nasopharynx swab or a sputum sample. Notably, viral RNA can be detected from stool from symptomatic and from asymptomatic carriers of infection. In this way, wastewater-based surveillance may be crucially beneficial, capturing the health of the entire community, including those individuals that may not seek care or testing, and capturing those individuals who may lack access to routine testing. Several studies have established that SARS-CoV-2 can be detected in wastewater influent and fecal sludge collected at the wastewater reclamation facility. And these studies have demonstrated that wastewater trends typically reflect those trends in reported clinical cases. In several incidences, increases in wastewater viral loads have preceded increases in clinical cases by up to several days. Wastewater monitoring in this way has become to be appreciated as a method for routine monitoring and can provide early warnings of potential outbreaks. With these benefits in mind, we set out to implement a monitoring program of our own here in Athens, Clarke County. We aim to better understand COVID-19 in our local area through wastewater surveillance. Ultimately, we hope to complement those surveillance strategies already in place in the local region, including randomized testing, clinical surveillance, and antibody testing. We're excited to share with you the results of our collections to date. First, we'd like to provide a general overview of our monitoring program. We've been conducting this monitoring program in Athens, Clarke County, a consolidated city county about 70 miles east of Atlanta. Athens is home to 124,000 residents, which includes 30,000 undergraduate and 9,000 graduate students who attend the University of Georgia. And while we appreciate the assets of our community, such as the mobilization of our research institutions and the medical trainees we have available, we must recognize some of the risk factors that make Athens, Clarke County particularly vulnerable to the consequences of the pandemic. In Athens, there are notable disparities in socioeconomic status and healthcare coverage, where 30% of our residents live below the poverty level and 20% of our community remains uninsured. A notable number of individuals live in group quarters, such as nursing homes or multifamily units. And thus, wastewater monitoring program here provides an opportunity to assess our community as a whole in a single sample. Athens, Clarke County has 3 separate water reclamation facilities that serve 3 unique catchment areas. In catchment area A, this area serves residents and a large portion of the industrial section of Athens, Clarke County. Catchment area A also includes the University of Georgia and its campuses. And the influent flow at catchment area A is the greatest of the 3 water reclamation facilities, with 5.32 average influent flow in millions of gallons per day. Catchment area B serves a large residential population of Athens, Clarke County, and serves the 2 hospitals that are located within Athens, Clarke County. It has a similar influent load of 4.8 millions of gallons per day. Catchment area C is the smallest catchment area in Athens, Clarke County. This catchment captures approximately 2 millions of gallons of wastewater per day and serves mostly a residential area in Athens. Of note, this water reclamation facility receives routine inputs from septic tanks and from porta potties throughout the county. Throughout this program, we have been sampling wastewater from water reclamation facilities that capture from each of these 3 wastewater reclamation catchment areas. We'll share here and now a little bit of our methods to show you how we examine these wastewaters for SARS-CoV-2. We collect a 24-hour composite sample from each water reclamation facility. We do this weekly or twice weekly. These composite samples are extracted for viral RNA using the QIAGEN viral RNA mini kit. The isolated and purified RNA is then converted into cDNA through reverse transcription using an in vitro reverse transcriptase. That cDNA is then analyzed using real-time PCR. We quantify the number of SARS-CoV-2 copies using the N1 and N2 gene targets. This gives you an overview of some of the controls that we use. We've been implementing bovine coronavirus, which is carried in calf guard, a vaccine, to estimate the recovery and inhibitions in our samples. Additionally, with every run, we run a positive control of SARS-CoV-2, a synthetic RNA produced by millipore sigma. Using qPCR, we set up standards using a bovine coronavirus DNA ultramer for our process and inhibition controls. And separately, we run a linearized plasmid specific to SARS-CoV-2 provided by IDT. Using qPCR, we target the M gene from the coronavirus. And for the target SARS-CoV-2 virus, we target the N1 and separately target the N2 genes. I'm showing you here our Lipp lab methods cheat sheet. So we summarize our methods here, and our methods are available and open access on our website as well. I'll share the link to these methods and to our data later on in this presentation. With that summary, we'd really like to share with you some of our preliminary results. To date, we have analyzed wastewater samples from all 3 facilities over a period of 35 weeks between June 30th to our most recent sample collected on February 24th of 2021. This is a total of 168 composite wastewater samples. For the first 18 weeks of this program, we collected composite samples once weekly. Then, in November 2020, we began sampling from each facility twice per week to improve the resolution of our data. We are representing in this figure here the estimates of the total viral load of SARS-CoV-2 in wastewater across Athens, Clarke County. The total viral load was estimated first by quantifying the number of viral copies detected in each wastewater sample from each facility, and then adjusting for influent flow at each facility. The total viral load is the sum of the viral load estimated across the 3 facilities. We present here the point estimates for the total copies of N1 and N2 quantified from our wastewater samples. Estimates for N1 are presented in green, and estimates for N2 are presented in orange. A lowest trend analysis was applied to these estimates separately, along with the 95% confidence interval for each trend line. While our 2 targets reflect similar trends over time, N1 has been detected at higher concentrations overall across the course of our study. The total viral load in Athens, Clarke County has ranged from approximately 10 log to 14 log copies and has been variable across the progression of the local pandemic. When we compare the temporal trends of our wastewater with those trends reported in clinical cases, a few patterns begin to emerge. Here, we're showing the number of newly reported daily cases in Athens, Clarke County, alongside the 7-day moving average of newly reported cases, that yellow line. I'd like to highlight some of the interesting time points and trends that we see in this data. This first mark here indicates when we had a large opening in Georgia, including bars and recreational areas. The second date that's marked here is the implementation of a mask mandate in Athens, Clarke County. And notably, this third date marked here is the first day of the fall semester of the University of Georgia. We see that a few weeks following the opening of the university, we do see the largest number of reported cases to date. If you compare the peak of this epidemic curve to the peak in SARS-CoV-2, you can see that they align pretty well. And notably, the highest concentration of SARS-CoV-2 to that point had been noted 7 days prior to the highest peak in cases. Now, as cases declined in Athens, Clarke County, we started to see a lot of variability in our data. At this point, we noticed that we actually had a lot of levels of non-detectable levels of SARS-CoV-2 in our wastewater. And so we see that as the incidence of COVID-19 cases declines, it's more difficult for us to detect SARS-CoV-2 in wastewater. However, after the holidays in November and December, we do see an increase in SARS-CoV-2 viral loads over time. And these continue to increase alongside increasing numbers of daily reported cases until this peak, when the peak in the number of daily reported cases coincides with another peak in the total copies of SARS-CoV-2 in our wastewater. We're approaching this period now where the number of daily reported cases in Athens County is declining, and there's been a lot of variability in the viral load of our wastewater samples. Overall, we have seen that levels of SARS-CoV-2 track readily with trends in reported cases, and that perhaps there may be some predictive power during certain periods, such as the beginning of the fall 2020 semester. But we do see some variability when case incidence is low. We're looking to improve our resolution with improved methods for viral recovery. Recently, and to date, we have been recovering viruses directly from wastewater without any prior concentration of our samples. To provide an estimate of our viral recovery to date, we've estimated the recovery of our surrogate bovine coronavirus process control. For each sample, we spike in bovine coronavirus, which is carried in calf guard, at a known concentration into our wastewater samples. We quantify the recovery through our extraction and quantification protocols, and here we demonstrate the final recovery of our process. In this figure, we present viral recovery across our collection period. Recovery is presented by each separate reclamation facility and is presented alongside the average recovery for that facility, which is represented by a dotted line. We estimate that overall across all of our samples, our average percent recovery has been approximately 49%. Between the three catchment areas, the average recovery is pretty comparable, 49, 53, and 43% respectively. However, we've recognized that viral recovery varies considerably across collection periods. We do not currently account for viral recovery in estimates of our viral load, but we do in the future intend to investigate differences in recovery based on physical parameters of the wastewater, such as chemical composition, total solids, and the potential impact of environmental variables such as rainwater intrusion. We intend to normalize our estimates in the future for recovery and total viral load based on additional biomarkers, such as chemical and other molecular parameters. Now that I've demonstrated some of the temporal trends, I want to share with you some of these spatial temporal trends that we've seen in our wastewater data. Here, I'm showing you again the map of the three catchment areas in Athens County. But now they're colored by an estimate of the total viral load per catchment area. And this ranges from 10 log to about 14 log of total coronaviruses. This is about a 30-second clip, so I'll take a minute and let you watch. But I'd like you all to notice or to make note of catchment area A, the catchment area here that's in orange, the one that serves the university, downtown, residential areas, and the industrial areas of Athens County. Now, what we find particularly interesting about the spatial temporal trends is that the total viral load of SARS-CoV-2 in catchment area A tends to increase prior to increases in the other two catchment areas. And we've seen this on 3 occasions. I'm showing you here the 3 accounts in which we've demonstrated that SARS-CoV-2 first increases in catchment area A, followed by the other 2 catchment areas. The first incidence of this occurred in early August, where you see an increase in catchment A, followed by the other 2. This occurred again in October. And then again in November. This has been really curious to us and something that we've been looking to investigate with more localized sewer line monitoring throughout the catchment area A and across the county. Alongside this research, we've been interested in sharing our data publicly and in real time. If you're interested in keeping up to date with the data that we're sharing, we're still collecting wastewater twice weekly from each of our water reclamation facilities and sharing that data online here at covid19.uga.edu/wastewater-Athens. And here you can look at our data as we publish it in real time. On this site, we simplify our data on the main loading page by taking an average of N1 and N2 targets. We report here on our website the average of those 2 targets, again with the low S smoothing trend curve and the 95% confidence interval of that low S trend. We always provide captions with our updates to give you our own account of how we predict or perceive changes in SARS-CoV-2 concentrations in wastewater and how those coincide with changes in cases over time. We'll be continuing this research and are interested in what we found so far and our future directions. Some of the key points that we've taken away so far are that temporal trends in wastewater tend to reflect those reported cases of COVID-19. However, there's decreased sensitivity of our wastewater monitoring during periods with low infection rates in the community. That is, we have a lot of non-detectable samples or samples where we cannot detect virus with our current methods. We've noticed that differences in spatial temporal trends between catchment areas may provide some evidence for localized hot zones or specific zones of localized transmission. In the future, we're going to be looking at the normalization of our SARS-CoV-2 concentrations with some outside biomarkers that are molecular and chemical. We will be analyzing and interpreting the actionability of some of the upstream sewer line samples that we've been collecting to see if we can really associate those with those spatial temporal trends that we've been seeing. Additionally, we're looking into sequencing our viruses detected from wastewater for applications in phylogenetics, and looking to quantify any new novel variants that we might be able to detect in SARS-CoV-2 wastewater. I'd like to thank you all for joining me for this presentation. And I'd really like to extend a lot of gratitude to our entire team, including our PI, Dr. Lipp, and the graduate and undergraduate students that have been working as a really great team throughout this project, including William Norfolk, Amelia Foley, and Megan Robertson. Many thanks to our supporters at in the Department of Environmental Health Sciences, our partnership with Athens Clarke County, our support from the University of Georgia's College of Public Health, the Center for Infectious Diseases, and for the Interdisciplinary Disease Ecology Across Scales program, the IDEAS program at the University of Georgia. If you have any questions or are curious about our research, please feel free to look at the website that is hosting our data, the covid19.uga.edu, the Lipp Lab website, or contact us directly at my email or the email of Dr. Erin Lipp. We look forward to connecting with you all in the future and appreciate your time and consideration. Thanks so much.